So we are a high throughput screening lab. We're a collaborative research facility. So you'll find that some of the things that you can find at our facilities, we have an experienced staff. So that's sort of the foundation. You heard about strong foundations earlier. We have a strong foundation. We have a good experienced staff, both in academics and industry, to help support your projects. We work a lot on assay development because all of the assays that come in need to do some development work so that they're compatible with the equipment and infrastructure that we have in our core. We do traditional high throughput screening methods, biochemical methods. We also have automated microscopy. So we can do a lot of the endpoints that were mentioned earlier this morning. And we're making a concerted effort into data mining as well. So project goals for a lot of the projects that we've worked on can really fall into these 4 different categories. So development of new molecular probes often take advantage of our small molecule collections. We do identification of therapeutic vulnerabilities. So these are more mechanistic studies. So they take advantage of our mechanistic informer sets of compounds that we have for offer. And then lastly is our repurposing effort, where we are looking mostly at drugs that are approved or investigational agents, either alone or in combinations. And there that's sort of to go with our secret support, so we're looking at translational endpoints there. And because we can do a lot of the imaging technologies, we have some toxicity, cellular cytotoxicity profiling that we can do as well. The types of projects that we run small scale. We can do proof of concept. If you need data to support a grant, we're more than welcome to work with you on doing that. We can do simple endpoints. Biochemical endpoints are fine. Those tend to have rapid turnarounds. But we've also worked on some fairly large projects as well, large panels of cells, complex cellular models, organisms based as well. We can do a lot of live cell screening and time-lapse studies that fit. These are just some of the targets that we put in. I had to put dengue virus protease in there for Suzanne. It was a project we worked on a couple of years ago. But again, biochemical endpoints have a quick turnaround. Our combination screens have been more translational in nature. We've had a couple of secret grant collaborations going in that area in particular. Right now we're working on the pediatric brain cancers you see listed there, and triple-negative breast cancer. And then some of the different types of targets that we've seen. We do work on molecular targets, toxicity testing, and then you can see in the advanced cellular models, some of the things, the more recent initiatives have been the 3D models and gels, so doing more organoid studies. These are from patient-derived cell lines. We have worked with zebrafish in some studies. In tissue chips is another area that we're starting to venture into. That's an NCATS-funded project that we have in collaboration with the folks in College Station, where we've been testing a variety of different tissue chips, but we're finding that some of these might be beneficial for some of the preclinical work that might be for new drug testing. And then lastly, the deliverables from the lab. Obviously, we'd like to deliver to you validated small molecule probes suitable to support your studies, and if we can, therapeutic leads, either alone or in combination, that could be useful going into animal models. So trying to help de-risk some of the combinations or drugs that you might take into an animal model. If we can work through the models and methods that we have to de-risk those for you so that you can have better chance of success, I guess, as you're going into your animal models because of the expense involved.
Thank you. Thank you, Clay.